How's it going, you guys? So in this video, I wanted to discuss signal transduction, the signal transduction pathway, as well as the reception of a signal, specifically in the context of protein receptors that use a G protein, so G protein coupled receptors. So that's what the topic of today's video is. We already discussed how cells communicate. They can communicate through cell-to-cell direct contact. They could also contact through chemical signaling, whether that be local regulators over a short distance or hormones all over a long distance from one part of the body to another part of the body through the bloodstream. So today I want to talk about what happens when that signal is received. So we are going to look at a specific signal transduction pathway that involves epinephrine or adrenaline. So we start off with a signaling molecule that we will call a ligand. So I'm going to point to this and label it a ligand. So a ligand is any signaling molecule that binds to a protein receptor. And this ligand is going to be the hormone epinephrine or adrenaline. Epinephrine. So this ligand will start off, our first step will be the ligand will bind to our protein receptor, our transmembrane protein receptor that spans the membrane. And so it has an extracellular side and an intracellular side. We say that proteins have domains, or just parts of a protein are considered domains. So this right here is the ligand binding domain. So step one, the ligand binds to the ligand binding domain. Now for step two, in the case of epinephrine, there is a conformational change, so a change in shape that occurs in the protein receptor. So the protein receptor, particularly the intracellular side, so we'll consider this part of the protein receptor the intracellular domain. So the ligand binds to the ligand binding domain. The protein receptor, specifically the intracellular domain of the protein receptor, undergoes a conformational change, a change in shape. This is coupled with this thing called a G protein. So we call this a protein receptor, this a G protein, this whole thing is a G protein coupled receptor. And so this conformational change in the intracellular domain of the protein receptor triggers a change in the G protein. And so the G protein dissociates, it splits off, where the alpha subunit ends up going this way. Now, the alpha subunit, once it's dissociated, once it's broken off, can bind to adenylyl cyclase, this new protein that's also embedded within the membrane, mostly on the intracellular side. And adenylyl cyclase, its job is to create a second messenger called cyclic AMP, or CAMP, C-A-M-P. And it does this by taking ATP, adenosine triphosphate, it removes two of those phosphates, creates a cyclical molecule, and now we have cyclic AMP. The M stands for mono, because we only have one phosphate. So just to kind of show this a little bit better, if we go over here, you can kind of get a better sense of how cyclic AMP is formed. So we have ATP. ATP loses two phosphates, and then this one phosphate ends up reforming a bond with this carbon of the central sugar molecule. And if you recall, the sugar molecule has carbons that we number in a clockwise fashion. So this carbon right here is the first prime carbon, 1 prime. This is the 2 prime carbon, 3 prime carbon, 4 prime carbon, and 5 prime carbon. And so this phosphate loses the two adjacent phosphate groups, and this phosphate reforms its bond with the 3' carbon. So what we end up seeing is a new molecule that only has one phosphate group, and it creates this cyclical shape that has been rebonded or bonded in a different way with the 3' carbon. Hence, cyclic AMP, cyclic adenosine monophosphate. So cyclic AMP is considered a second messenger. So let's just do a quick recap before we move on. So let me see if I can get everything in the picture. Let me lower this down a little bit. Maybe that'll help. There we go. And there we go. So as a quick recap, the ligand, in this case epinephrine, binds to the ligand binding domain. This triggers a conformational change in the protein receptor, particularly the intracellular domain of the protein receptor. This in turn will cause the G protein to dissociate. The alpha subunit will leave the beta and gamma subunits of the G protein. It will go to the adenylyl cyclase enzyme. Once it binds to that, adenylyl cyclase will be activated and it will start converting ATP into cyclic AMP. And to kind of connect ideas, ATP is coming from cellular respiration, whether that be glycolysis, the Krebs cycle, or the electron transport chain with oxidative phosphorylation. But we have cyclic AMP, which I will represent as a triangle from this point forward. Now, this triangle, the cyclic AMP, is now going to trigger a cascade of responses. So I'm going to say that this right here was step 2, the conformational change in the protein receptor with the G protein, dissociation, all that stuff. And then once cyclic AMP is generated, we can now say we are entering step 3, where the signal will be passed down and it will be amplified, which we'll talk about in a second. So cyclic AMP is generated. It binds to this new protein, protein kinase A. One thing that you should note about kinases, they phosphorylate stuff, which means they add a phosphate group to something new. So I'll put over here, kinases phosphorylate stuff. So we have protein kinase A. So if it's a protein kinase, it phosphorylates some other protein. If it's a phosphorylase kinase, it's going to phosphorylate a phosphorylase enzyme. So if it's a kinase, it phosphorylates something. Now in this case, pKa, or protein kinase A, is normally in its inactive state with 4 subunits. 2 of those subunits inhibit the 2 catalytic subunits. And that's because these 2 subunits are blocking the active site of these 2 catalytic subunits. So what happens is CAMP, cyclic AMP, binds to these secondary locations called allosteric sites. I'll put that over here. Allosteric sites, or just site, allosteric site. So we have 4 allosteric sites here. Cyclic AMP binds to those sites, triggering a conformational change in the 2 inhibitory subunits. That will, once it changes shape, it will release these 2 catalytic subunits, and these 2 catalytic subunits will become active. So we've now activated protein kinase A. We've caused the CAMP to bind to allosteric sites, causing the whole molecule with its 4 subunits to dissociate, and now we have these 2 active catalytic subunits. Now, these subunits will work as kinases. They will phosphorylate stuff. These subunits will go to a new protein. This new protein which is phosphorylase kinase. This new protein, in its normal inactive state, will then be phosphorylated by the catalytic subunit. It'll be phosphorylated using ATP, so we'll take ATP, take off a phosphate group, make it ADP, adenosine diphosphate. This extra phosphate will bind to, again, an allosteric site, allosteric binding site. Once it binds, it will activate, so I'll put activated. It will activate PHK, or phosphorylase kinase. And this will then do the same for the next enzyme. So GP, or glycogen phosphorylase, will then be phosphorylated by this kinase. It will go from its inactive state to its active state, because now it has an extra phosphate group in its allosteric site. And once that is activated, glycogen phosphorylase will trigger the cellular response, which in this case is to break down glycogen. So glycogen, if you recall, is a polysaccharide. It's made up of many sugars. It's this super branched structure. So we're going to break it down to release sugar, because epinephrine, adrenaline, if you recall, kind of triggers this fight or flight response, which requires energy. So epinephrine, long story short, ends up leading to this cellular signal to break down glycogen, to release sugar, to break that down even further, to make energy for a quick release energy situation. So, all together, step 1, ligand binds to ligand binding domain. Step 2, the receptor undergoes a conformational change on the intracellular side, the intracellular domain, triggering the production of a secondary or second messenger. And then this second messenger, for step 3, will then trigger a new cascade of signals. And then finally, step 4, we get our cellular response. So that is the signal transduction pathway. This is a very complicated signal transduction pathway, but this is an often cited pathway with epinephrine. So if you know this, it'll be a lot easier to understand other signal transduction pathways. So hopefully that was helpful. The last thing that I want to mention, some just final notes, is this whole thing where we phosphorylated this protein, and so once that phosphorylated kinase is active, it will phosphorylate a new protein. This whole process is considered a phosphorylation cascade, where one protein gets phosphorylated. It becomes active, phosphorylates a new protein. That protein or enzyme becomes active, and that phosphorylates a new protein, and so on and so forth. So that's pretty much it. Oh, last thing. In terms of understanding the naming scheme, this will help you remember what certain enzymes do. So kinases, as we say, phosphorylate stuff. A protein kinase will phosphorylate a protein. So protein kinase A phosphorylates this protein, and phosphorylase kinase, a different enzyme, will phosphorylate a phosphorylase, which in this case is glycogen phosphorylase. Sorry, this should actually be GP. Glycogen phosphorylase. And glycogen phosphorylase, once it's phosphorylated, becomes active, and it will break down glycogen. So that's just some elaboration on the naming scheme here to help you understand what each enzyme does and why it's named the way it is. So hopefully that was helpful, and I will see you in the next video.